Good afternoon, everybody. Let us start today's webinar. We are extremely privileged to have as today's speaker, Dr. Suman Kapila from National Dairy Research Institute, Karnal. Dr. Kapila did her PhD in animal biochemistry from NDRI, the same place where she is serving as principal scientist now. Dr. Kapila has 27 years of research and teaching experience, and she has guided 14 MVSC, MSc, and 13 PhD students, which include 2 international students. Currently, 5 PhD students are pursuing their doctoral program in her laboratory. Dr. Kapila is extremely productive researcher, evidenced from her total number of research paper at total number of research paper being 160, and she has 2 patents. And today she will talk on a very interesting topic on the role of milk-derived peptides as potential osteoanabolics. We look forward to hearing very interesting and exciting data, Dr. Kapila. Now, the webinar stage is yours, please.
Yeah, thank you, sir. At the outset, I would like to thank Dr. Navidya Chattopadhyay for giving me this opportunity to share my little work. I say the amount of work you do with regard to bone health, and that comparative to that, my work may not be that good. But whatever the work I have done, little work, I'm going to share. So first of all, I would like to tell you about the National Dairy Research Institute. The name itself is clear. We are working only on the dairy side. So that is the milk. So we have basically 3 divisions in NDRI. The workers that are working on production, processing, and the management. So different scientists, they are working in these 3 categories. And my area falls within the processing group. So basically, our mandate is to improve the value of the milk, to make value added products, to give the to do research in milk, to establish its health benefits scientifically. So keeping that in view, I have started this work. So today I'm going to talk about milk-derived bioactive peptide as potential osteoanabolics. So as you all know, milk is a nutrient dense food. That is, any food which is called nutrient dense means it has very good amount of nutrients with respect to the calories it is providing. So in that way, it is the best food you can say. So basically, if you're talking about milk-derived bioactive peptides, we have to move to the milk protein. Basically, milk proteins are of 2 kinds. One is casein and second is whey protein. Casein constitutes 80% of the milk protein and the 20% is the whey protein. So these are the structures which are shown. This is the casein micellar structure, which is the casein is present in the form of casein micellar structure, and this is responsible for the whiteness in the milk, because when light scatter to it, that why you get this white color. And this is very unique molecule, and this is the protein which don't have a very tertiary and quaternary structure. It's a simple protein. Secondary structure you can see in this. And then you have beta lactoglobin and alpha lactalbumin. These are the 2 major whey proteins, and they are nutritionally very important protein. And you know, the whey supplements are usually taken by our athletes, and the gym also they prescribe. This is because of the composition of amino acid. They have good amount of branch chain amino acids, which is very good for muscle health. So now moving to the importance of milk protein hydrolysates. Milk protein hydrolysate, they are obtained from the protein after the enzymatic digestion. So what was the basic purpose to have this hydrolysate? Because there are certain components in the milk which show allergenicity to certain individual. So the basic purpose initially it was to decrease the allergenicity, because in the milk it is the beta lactoglobulin which is mainly responsible for the allergic reaction. If certain individual are allergic, it is because of that only. And then to increase the absorption, that is the another important purpose, and then to improve functional properties of the proteins, because we are working in a dairy institute, so we are associated with the formation of so many products, different kind of cheeses. So for different kind of products we need different kind of protein texture. So that basis we make milk protein hydrolyzed by using enzymes or bacteria and so. And when these milk protein hydrolysates they are obtained, they have a very good amount of bioactive peptides. Now the question arises, what are bioactive peptides? Biologically active peptides or functional peptides, they are the food-derived peptides. They have their nutritional value because they are again the polymer of amino acids, so amino acids are there. So from that point of view, they have nutritional value. But apart from their nutritional value, they have some physiological effects in our body. And these bioactive peptides, till they are the part of the protein, they are inactive. But once they are released by the action of different enzymes, by bacterial enzymes or digestive enzymes, so these bioactive peptides which are generated, they have some physiological role and they may act like a hormone sometimes. So if you see in general, the bioactive peptides usually contain 3 to 20 amino acid residue per molecule, and these are, I have shown in this slide how to get this bioactive peptide. I talked about casein and the whey protein, and then how we can generate different kind of bioactive peptide by using these different ways, by using starter culture, by using probiotics, by using proteolytic enzyme or the digestive, either by in combination or individually. So you can have different kind of bioactive peptides, and these bioactive peptides have been studied for all these properties. ACE inhibitory have ACE inhibitory property, they are antioxidative in nature, they have antimicrobial activity, immunomodulatory, and mineral binding. Opiates are like this. And in this you can see the list of major bioactive peptides which are present in milk. If you see most of the bioactive peptides initially I have mentioned, they are opioid in nature, and usually they have tyrosine at their N-terminal end. So there are certain basic features. If it is ACE inhibitory, proline may be there. If it is immunomodulating, other amino acid sequence. So in this basis, lot of bioactive peptides that have been characterized and their functional role has been attributed. So when I was when I started this work on osteoanabolic peptides, I was working with ACE inhibitory, antioxidative, and immunomodulatory peptides of the milk. So then then I think it was in year 2010 when I met Dr. Ritu Trivedi, and she was discussing about the osteoanabolics. Then I thought of, why don't we try this our peptides, whether they have this osteoanabolic or osteogenic activity or not. And we started from there, I think in 2010, when for the first time I visited a CDRI. So from that day onwards, we have done a lot of work on these bioactive peptides. If you see the milk-derived bioactive peptide in literature, you won't find much work, only except NERVA work. That was the lead which we get when we have started our work. So they have done the role that this bioactive peptide, they are the osteogenic peptides, the peptide tripeptide IPP and VPP. Therefore, the first time they have reported. Then we have started working on our milk, that is buffalo and whey peptides, and we have characterized these peptides. I will explain how we have carried out this work, and then we have modified also some of the peptides. So basically, why we have started work? Because we have a problem of osteoporosis that you all know, and no need to tell what is osteoporosis. But according to WHO, osteoporosis is characterized by low mineral density with microarchitectural deterioration of bone tissue, leading to enhanced bone fragility and increased susceptibility to fracture. So if you see the risk factor of osteoporosis, it is the inadequate intake of calcium, and you know milk is the richest source of calcium. Moreover, the bioavailability of calcium from milk is the maximum. It is the ratio of calcium and phosphorus. Any product, if you have the optimum ratio, only then the bioavailability of calcium will be better, and milk is one such product. Then deficiency of vitamin D. This is another very important reason. And though milk is not having good amount of it, then we have poor protein.
Please accept the speaker. Everybody should mute. Everybody should mute except the speaker. Okay, sorry. Please go ahead.
Yeah. Then poor protein intake, endocrine defect, hyperparathyroidism, PTH. The role of PTH is very well known in calcium homeostasis. How whenever the calcium level falls, how it stimulates the PTH, and then PTH in turn convert the vitamin D to its active form, and then vitamin D bring about a lot of function to maintain the calcium level. And if it is high, then how it causes the release of calcium from the bone, that you know. Then estrogen deficiency, that is the major cause of osteoporosis, postmenopausal osteoporosis. And then problem is there, then certainly solutions are also there in the form of drugs. And whatever the drugs that are there, that are again categorized into two: anti-resorptive agent and the osteoanabolic agent. Anti-resorptive, you all know, alendronate, then lasofoxacin, that is the receptor binder of the estrogen. It binds to estrogen receptor, mimicking that the function of estrogen in that way. But then you have osteoanabolic agent. So all these anti-resorptive osteoanabolic agents are there, but even why there is need for other molecules, this is because they have many of these drugs, they have many side effects. So then, since milk is always milk is always considered good for bone health. So keeping that in mind, we have started that why not the apart from calcium, apart from the bioavailability of calcium, there must be something more in the milk which is causing the improvement in the bone health. Then we start looking for the bioactive peptides. So how bioactive, that I have mentioned that milk is the source of calcium, and the bioactive peptide, how they can enhance the bone. With this, we have started our research, and then we have started looking in the literature. So then we have find that in epidemiological study also, it has shown that milk consumption in childhood and during adolescence is related to high bone mineral density in adulthood. Then the studies have been conducted in pre and post menopausal women. They have shown the good effect of milk and milk-derived products. And at the same time, the lot of work being carried out in the area of bioactive peptides. So we started looking for that. And this is the very first study which we carried out. Firstly, we have used the casein, and its hydrolysate was prepared using trypsin. And these hydrolysates were used for the see the effect on the osteoblast proliferation and differentiation in mouse bone marrow culture. So this was the first study which we had, and this study has given us, you can say, lead that yes, casein hydrolysate, they are improving the proliferation as well as the differentiation of the bone marrow culture into the osteoblast. But to do the culturing every time and putting different hydrolysate was a difficult task. Then we started looking for some other ways, whether some other peptides which we have, whether they can be used as osteoanabolic peptide instead of searching each fraction and studying its osteogenic role. Then we find yes, ACE inhibitor, these are the findings that ACE inhibitor reduce the risk of fracture or improve bone metabolism on elderly people, and oxidative stress that also inhibit the osteoblastic differentiation of bone. So can we use ACE inhibitory and antioxidative peptides which we have for this osteogenic study? Because to do the ACE inhibitory assay and antioxidative, that was easier for us. You can do in a simple spectrophotometric assay, and you need not to go for culturing every time, because culturing to the mineralization, say 21-day culturing, that was a tedious task. So then this is the strategy what we have followed for digging out for the osteogenic peptides. So what we did first, we have isolated the casein by acid precipitation method. If you reduce the pH of milk to 4.6, what you get is the paneer part, that is the casein, that will be separated, and that then it is treated with different digestive enzyme, pepsin, trypsin, chymotrypsin, and its combination, and we get the hydrolysate. And these hydrolysates were checked for ACE inhibitory and antioxidative activity. And these hydrolysate, then we checked for this. We have cultured the calvarian cells, and in that also we have seen the osteoblastogenesis and osteoclastogenesis, whether it is affecting that way or not. Then what we did, we have, both of us having this casein hydrolyzate, we have started fractionating because we want to end up to the bioactive peptide level. So we have passed this casein hydrolyzate to 10, 5, 3, 1 kDa this membrane filters, and then size evolution chromatography, and then reverse phase HPLC. At every stage, we did the ACE1 and that is ACE inhibitory and antioxidative check activity, and then the last the fraction, which the fractions we got in and which was checked for this activity, the fraction which was showing the maximum activity, that we sent for LCMMS, and from that we got the list of peptides, and that 15 peptides, and out of these few peptides that were synthesized, and that peptides then again checked for all these activities. So this is the separated casein, how we have separated the casein, and then we have studied the duration of hydrolysis, for how long we have to hydrolyze. Then we have checked the this is the ACE inhibitory activity, and this is that is antioxidative activity, which is expressed in terms of TEA, that is Trolox equivalent, and this is the ABTSSA. And if you see, we have used different combination, and we found that the pepsin trypsin hydrolyzate was giving us the maximum these activities. So then we focused on only on pepsin trypsin hydrolyzate, and that hydrolyzate was made to pass through this different membrane, and at the end we were having 4 retentant and 1 permeate, and these were all again checked for this activity. And in this we found it is the 1 kDa fraction that was showing the maximum activity, that is the ACE and the antioxidative. Then it was made to pass through this HPLC column, and then we collected the different fractions, and again we have checked the this activity in this fraction, that is ACE inhibitory and the antioxidative activity. And then if you can see over here in the fraction 11, again activity was high, so we were restricted ourselves to this fraction, and then these were the this was subjected to LCMS, and then the peptides, 15 peptides we got in this, from alpha S casein, alpha S2 casein, beta casein, and the kappa casein. These are the different 4 type of casein which is present in the milk. So from here, then we have to choose one peptide from each. So we have chosen the smaller one, keeping in view its bioavailability issue. So we have selected this nonapeptide, then heptapeptide, and again this octapeptide from these, and they were custom synthesized, and then they were studies about the antioxidative and ACE inhibitor that we have. And then we have started working on the osteogenic effect of these 4 peptides which we have selected. Now, for osteoblastogenesis, you all know that first step is the commitment, then proliferation, anything which helps in proliferation of pre-osteoblast to osteoblast, and the differentiation of it, and then the mineralization. So all these stages it has to come across, or it has to go through all these stages to say that it is having some osteogenic growth. So on that basis we have started our work. First we have seen the effect of all these 4 peptides in different concentration on the proliferation assay, simple by MTT assay, and we have chosen certain genes also of the cell cycle to see their effect, then on the differentiation. So these are the 4 peptides, and in addition we have chosen the bone forming peptide and IPP, which was the already known in the literature that they have the effect, osteogenic effect. So on that basis, these are the markers COL1, alpha, OCN, and ALP that we have selected. And this is the mineralization finally, whether how these peptides they are helping in the mineralization. So from this study we found that peptide 2 and peptide 3 they were showing the best effect. Then this is the result of peptide 2. So one of my students was working on this, then he wanted to know how, if this peptide is causing the osteogenic effect, what could be the reason and what could be the mechanism. So based on using 2 inhibitors of AKT and ERK pathway, he has then carried out this work, and he has found that it is the AKT derived pathway that this peptide is working, and it is helping in the osteogenesis. Then this results we have published in Journal of Nutrition. Then before going to the in vivo study, we thought that since we have to give this peptides orally, this is our aim. So first check its transport. So then we did the transfer assay. This, you know, this is the transfer well-known assay. The Caco2 cells, cancerous cells, colon adenoma carcinoma cell lines, they were cultured, and then our hydrolysates, that is the peptides, synthetic peptides, which we were having, that were put into it, and after different time interval, we will see that how much of it is coming. So on that basis, we have studied the transport or viability of these peptides. So while checking this, we have chosen two positive peptides, positive which are known to pass through the transfer assay. One is bradykinin and another is beta-casomorphin. So these another two peptides we have chosen, and we have carried out this study. Since our peptide is not something that we can estimate as such whether it is there in the solution, so we have to run HPLC every time. So initially, first before starting the work, we have see what is the retention time on HPLC, and it comes around 21 minutes. But when we have added this peptide to the Caco2 cells, and Caco2 cells have proteases, immediately after adding, we got a second peak. So we thought our peptide is gone. So now after 60 minutes again, when we saw, it was there, but to our surprise, both the peptides, they were reaching into the basal chamber. So our peptide, though it is converted or it is broken down product, is also there, but both products, they were reaching the lower chamber. So then we have started working on the saturation concentration and the saturation time to find out how much concentration we have to use in this and how much the time we have to carry out this experiment. So this for bioaccessibility study. So this is the parameter that we have standardized, and then we have found out that our peptide as well as broken down product, both are coming into the lower chamber. So bioaccessibility is there. Then we started looking for if it is there, what is the route. Then we use for transport, different routes are there: paracellular transport, then you have receptor mediated transport, you have endocytosis. So we have used inhibitor of these. So these are the Wortmannin is an inhibitor of endocytosis process, cytogelicin is an inhibitor of paracellular transport, and glycine proline is an inhibitor of peptide transporter, which is this is glycine proline. It is a transport shorter peptides of 3 to 4 amino acid length. So that was used, and when we carried out this study, we found it is the peptide transporter that is affected when we use this, and the transport was reduced, and both in case of intact peptide as well as in broken down fragment. And similar study, similar effect we have seen in BCM5 also. That also breakdown fragment was also seen there. And in this case, the Wortmannin, that is the endocytosis process, that was inhibited to some extent. That is showing that this may be the pathway. So based on this, we found that our peptide, which is a heptapeptide, it transported in the intact form only to at the level of 1%, and broken down fragment at 1 to 1.16% in that way. And if you see the route of transport, it is peptide. I told you that peptide is usually for shorter peptide, 3 to 4, but our peptide was 7 amino acid long. So we were not very sure that this could be the pathway, since nowhere it is mentioned that longer peptides up to 6 or 7 can be. So then we come across another transporter, SOPT2. So in the literature, though we could not confirm it whether it is SOPT2 or peptide, but our study gives suggestion that it can be either of these transport pathway which our peptide is following. And after that, we had moved to the in vivo study. We have carried out the help of CDRI, and then we carry out this study in OVX rat, and in this case we have given our peptide at 2 concentration to the rats, 50 microgram and at 100 microgram per kg body weight, and the positive control in this case was a PTH based on literature, that is 30 microgram per kg on every alternative that was given. And this study was carried out for a period of 8 weeks. And then these are the results which we got in the sham group. So there is no change in weight, but in the OVX it was obvious that there was increase in weight was observed, but on intervention, when peptide was given, we have given at two concentration, 50 and 100. We could see the effect, though not as good as the PTH, but it was there. And whenever this ovarectomy is done, uterine cavity, that is, there's a regression and degeneration that takes place. So that cavity reduces, that you can see on this picture. Then bone turn, the effect on bone turn markers and biochemical strength that was seen as expected whenever ovarectomy is done, that the level of osteocalcin and ALP activity that enhanced. But on intervention, when these peptides were given, there was drastic reduction we could see here. And then the stiffness of the bone improved, and as and the energy to break the bone also improved, that you can see over here. This is the micro-CT images that we got from CTRI. Our students went there and would have done this work. So this is the images of the ovaries, and this is when the peptide given at 50 and the 100 micro. So certainly improvement was there, that could be seen. And then these are the different markers, the tubular micro architecture, that improvement was seen. Since our peptide was initially an antioxidative, right? So wanted to see whether there is effect on the oxidative stress. So in this case, we have measured the MDA level, which is the lipid peroxidation marker, and the GSH level, the antioxidant. So in the weeks there was increase in the lipid peroxidation, but on intervention there was reduction, and similarly GSH level also improved, and catalase activity improved to some extent, and SOD also. So antioxidative activity, enzyme activity also increased. That then effect on the bone resorbing cytokines. So Rankel, the pro-inflammatory cytokine interleukin 6 and TNF alpha, their level increases in OX group, and but when the peptides were given and the PTH, there was reduction. And TGF beta, which is a regulatory hormone, regulatory cytokine, sorry. So when its level decreases, but on intervention there was improvement. So in nutrition, and Dr. Ritu Trivedi lab, they have helped us to carry out this work. And then another study we have carried out on whey protein. Like casein, we have not started from hydrolysate this time. This time we have started looking for antioxidative and ACE inhibitor peptides, which are already known in the literature. And from there we have selected 6 peptides, 2 antioxidative and 4 ACE inhibitory. So first they were selected on the basis of all in vitro studies on colloidal cell culture, on the pre-osteoblast to conversion of osteoblast, and then mineralization. So on that basis we have selected 2 peptides. One is antioxidative and another is angiotensin, that is you can see for pentapeptide, and this is the 4 amino acid peptide that we have selected from the whey protein. And again the similar kind of study we did, and these are the micro-CT images of the result that we got, that when antioxidative and ACE inhibitor peptide from the whey source, whey protein, they were given, we could see the improvement in the bone architecture. And in this case what we found, that the antioxidative peptides, they slightly perform better than the ACE inhibitor peptide. This was the study that we, these are the 6 peptides which we have selected. I was talking about from the literature, and from this 4 we have selected these 2 and carried out the whole study. Now the last part which we have done recently, that is the microRNA delineation during osteoblast differentiation, which is induced by our heptapeptide VLP-VPQK. So we have started carrying out, we have done the microRNA profiling, then we want its target, and then the validation of the target under in vitro, and then in the in vivo study. So this is the plan which we did. We have used the osteoblast cells, and then to that we have added the peptide C initially, because 4 peptides were there. So this is the peptide C, that is heptapeptide. So it was added at a concentration of 30 nanogram. So this is the concentration we have used in all of our experiment, because at this concentration it has shown us the maximum effect. And this profiling was done after 4 days of with this Pepsi, and when what we got is the miRNA profiling 191 miRNA, and selected the miRNA by bioinformatics. In silico screening, target prediction was made, and then miRNA validation was done, and then in vivo characterization of selected miRNA that I'm going to tell you. This is the activity one before transfection. We in the validation part we have done two activity before transfection and after transfection studies. So this is the miRNA. We have selected the selection sorting was done in 3 steps. Sorting were initially what we got is 191 miRNA, and out of this top 20 down regulated and top 20 up regulated they were selected, and then identification of homologous miRNA by BLAST was done, and then based on the complete seed match and high similarity scores and validation on the target that was identified, and in the third sorting step we have looked for the novelty, and then mRNA with the validated target, and miRNA whose mode of action is less known. So based on that we have selected out of 191 we have selected only 5 miRNA 300, miRNA 592, then 381, 109 and 369. So I'm going to tell you the work of miRNA 300 over here. So you can see in this slide, this is the miRNA which was down regulated when Pepsi was given. So we wanted to check the result which we are getting in silico study, whether the similar result we are getting in vitro. So when this Pepsi was given, we see the expression of miRNA. So it was significantly down regulated. In this case we have used another peptide positive control BNP2. So in that too also there was suppression of miRNA3, and then through the target identification we found that this miRNA3, the target for it is SMAD3 based on bioinformatics tools in silico work. So we wanted to see the effect of a peptide on this SMAD3, and what we found that our peptide C, it significantly increased the expression of SMAD3. Then we have transfection study. So transfection was done same, the 3 sets are always run, the miRNA control, mimic and the anti-miRNA, that is the miRNA inhibitor. And then when these were in transfection was carried out, so we know that when mRNA is down regulating, so it is suppressing the ALP, and anti-mRNA will increase the ALP. Before going further, we have standardized the concentration of these mimics and the anti-mRNA inhibitor. So these 3 concentrations we have selected, and based on that these are the results. You can see when these 3 concentrations were selected, the decrease in ALP activity was significant, as this in the presence of mimic, and anti-mimic causes the significant increase in activity at 50 nanomolar, and in presence of me that you can see the different osteogenic genes, biomarkers genes, there was significant reduction at 50 nanomolar concentration, and in anti-miRNA in presence of this. So though effect was seen as 20 also, but significant effect we would see at 50 nanomolar concentration. And just to see when we are adding mRNA, what is the status of mRNA. So naturally when we are adding more of it was seen, and more of miRNA means there is reduction in the anti-miRNA that we could see over here. So based on this 50 nanomolar concentration was selected. Now we have to validate the target, and MIR 300 target is SMAD3 based on the homologous. So we have done the dual luciferase assay. So in this case 4 transfections were carried out, and we have made 2 constructs. One was having 3' UTR SMAD and other was not having. So in this way we have carried out this experiment, and the result we got, you can see over here that the suppression that the luciferase activity suppressed when MIR 300 was present in higher amount, so you can see. But on the other hand, there was no change in the relative luciferase activity. And then we wanted to see the SMAD level also in presence of MIR, and as expected, there was a significant down regulation in presence of MIR. The SMAD significantly down regulated, and so it also showed that the target for our MIR 300 is SMAD. Then we, since we wanted to see if MIR 300 is there, what is the effect on the differentiation? So this is the concentration we have selected, 50 nanomolar for 4 days treatment. Then we see the effect on the ALP activity and the collagen, also ALP staining and the collagen staining. You can see there was, when MIR was added, there was decrease in the stain. And then at the protein level, the two proteins, that is osteocalcin, two biomarkers, and this PINP, their level were checked, and in presence of MIR3 there was a significant lower level of this osteocalcin and this PIMP were found. But when anti-MIR 300 was added, there was a significant increase when it was. So then at the mineralization level, again in the presence of MIR 300, there was inhibition in the mineralization, but when anti-MIR was given, there was stimulation, or you can see that good mineralization was seen. Now we know that MIR 300 is working through SMAD3, and MIR3 is also causing the effect on the osteogenic differentiation. So how to link from MIR to genes? What is the complete pathway that we wanted to work out? So on that basis, then we have started studying the literature, and we found that SMAD3 works through beta catenin, and it causes the mobilization of beta catenin to the nucleus, and there it controls that RNX2, which is the main bone transcription factor, and then the which enhance the bone functioning and the differentiation. And this also we have validated from our in silico study by string model and enrichment study, and we can see the network is a strong network connection between RNX2, SMAD3, and the beta. And this is the study shows that MIR 300 targets and regulates the SMAD3 and regulates the downstream signaling both at the gene level and at the protein level. You can see, so whenever MIR3 is added or transfected into the cells, so there was significant reduction in beta catenin and RNX2. So it means our miRNA is working through this pathway, and when anti-miRNA 300 was given, there was significant upregulation. Then the similar result was seen at the protein level also. You can see there was reduction in the RNX2, beta catenin, and SMAD3 in presence of miRNA 300. So then this is another study which we have carried out, that what is the effect of this MIR 300 on the different osteogenic genes. So that we have already studied also. So again, we found in this case also there is a significant reduction of all the genes which are related to the osteogenesis on the well-known markers, and then also the SMAD3, beta catenin, and RNX. So the story is complete, that from SMAD, beta catenin, RNX to the osteogenic gene, this pathway, our miRNA 300 is working. Then from in vitro study, we move to the forget to tell you, this study is on rat pups. Actually, I'm not able to see the title because of some presenting way. So this study we have carried out on the 2 days rat pups, and in that this miRNA and anti-miRNA that was given, and then the effect was seen. So this is the extension of in vitro study to ex vivo study, and from ex vivo study we move to the in vivo study, and this study we have carried out in ovic rat, and then again same MIC, MIR, and anti-miRNA that was given weekly one injection and 30 at the rate of 30 microgram. And then after the study, the this micro-CT that was done, and you can see in presence of MIR-300 there's a deterioration of the bone architecture, and that is all this bone volume, connective density, tubular number, BMD, and but anti-miRNA 300 has improved the bone health or bone architecture that you can see over here. And then if you see the expression of the bone itself, then was SMAD3, beta catenin, and the likes, they were completely down-regulated in this case. That was the study about MIR-300. Similar study we have carried out. I'm just simply running similar kind of study we have done. In this case, the target, in that case, SMAD3, it is BMP27. So again, similar way, we have checked the level of BM2 and PEP-C, and to see the expression by adding this to see the expression of MIR-15. Then we have just one. We found that MIR-592 negatively influenced osteoblast differentiation and mineralization. Similar way, this is the complete network how BMP2 is causing the that, because MIR-59 is affecting the BM2 and how BMP is passing the signal. So this is the signal cascade, and we have seen all the expression of this MIR-59 and NTM on all these molecules. BMP7, BMP-PR receptor 1, receptor 2, SMAD1, 5, 8, SMAD4, 6. So, on silico analysis also we found this network, and then carried out study again in I think PEPs similar way, and we got the similar kind of result. And if we found that it is the phosphorylated SMAD that is inhibited in presence of MIR-592. The study we have carried out again in rats. See that MIR-59 causes the deterioration in the bone health by increasing the reducing the density of the bones, as you can see in the bone mineral density data. And similarly, we have seen the different markers that the signal transduction pathway that were also checked. There is no effect on 1,5-T, but on the phosphorylated SMAD we could see, but SMAD4 and SMAD that were affected. So this is the overall study. So our study that has shown that peptide C, that is VLPVPQK, that is the haptapeptide, it has the ability to regulate the expression of miRNAs, miRNA 300 and miRNA 592, both were down-regulated, and thereby it checks the expression of genes and targets SMAD3 and BMP7, SMAD158 and NS2, which are the indispensable for the bone regeneration. So PEP-C is, you can say, peptide which is stimulating the bone health or the osteogenesis by down-regulating the miRNA 300 and miRNA 592. So then we have carried out one more study with our peptide. So we know the sequence of our peptide. Then we have started looking the similar kind of sequence in other species, in goat, in other wild animal also. So on that basis, we have modified our peptide, just a single amino acid change, and we have generated this 15 analogs, you can say. They were differing from the original peptide only by 1 amino, 14 we have prepared. One is our original peptide. So, and these all peptides then were checked for their osteogenic effect by studying on all the activity of the ALP, osteocalcin, and bone mineralization. All that data was generated, and on that basis we found that peptide C, this is on the principal component analysis, we have screened because effect was not that much significant. So but on the basis of this principle, we could make out that the peptide 7 has given us the best performance among all the 14 peptides which we have generated. So then these two peptides were compared for their effect on the all osteogenic genes, that is ostrix and OPG, BMP2, RNX2, and we can see that there is a effect of, this is our original peptide and this is peptide 7. In that peptide 7 at third position, proline is changed via tyrosine, that is VLP, it is VLY, the rest of the sequence was same. So there was improvement in the activity of all these genes, and still we are doing the in vivo study related to this peptide, that is still going on. So at the gene level and at the protein level, we could see the, this is the major finding of what other study we have done in the last 10 years. So we have characterized 4 osteogenic peptides from casein, 2 from whey, and that from there we have worked out their transport study. Then we have done that they are bioaccessible, and then we have carried out their how their mode of action, how they are showing the osteogenic effect through their miRNA, different miRNA regulation, and one peptide through AKT pathway that we have worked out. And since these peptides were antioxidative, so we have found also that their antioxidative action is clear-cut shown in the in vivo study, and we have applied for a patent, and patent is almost granted, plus one it is subjected to NB approval. I think that has also been done, and in a few days we may get this patent also. So at the end, I would like to thank you all who have contributed to carry out this work, and our director MS Chauhan, Dr. Rajiv Kapila. He and me, both we are working together. Although he is more focused on the probiotics, I'm doing more work on the bone health. Then the team of our CDRI who have helped us every time when we need their help. Dr. Navedya Chattopadhyay, Ritu Trivedi, and Divya Singh, thank you so much. And all my PhD and the MSc scholar. Thank you.
Okay, an impressive piece of body of data that has been accumulated over a period of a decade.
10 years.
Very commendable work. Now, I have a few questions.
Yes, sir.
Number one is that since you have found that MIR 30 bay, 300 bay, 300 targets MAG3, and you have studied the downstream of that MAG3, which of course beta-catenin and RUNX2. But one of the major regulators of MAG3 signaling is TGF-beta pathway. So, do you think that your peptide is somehow also regulating TGF-beta pathway?
So I can't say right now, but we can see, we can try. So we have not looked for that. TGF beta.
Yeah, because you have done nearly almost close to your presentation, you have shown that BMP7 regulating SMAD158, and then 2, 3 are generally regulated by TGF-beta, the major regulators.
Yes,
And
yes, yes.
you know that TGF-beta also has a very profound action on bone cells, not only in osteoblast, also in osteoclast. And in osteoclast, it will dedicate that it can activate osteoclast function. So we may try to see it. You may consider.
I have, yeah, go.
Yeah, please. Yeah.
We have initially we have worked on osteoclast also. Initially when we have started, somehow we could not carry out that work because of some problem. But we see that our peptide was decreasing the osteoclast number as well.
Okay. Yeah. Okay. Another thing is that in one of your slides, I have noticed that you have compared, when you are comparing the peptide with PTH, you showed that it doesn't have any uterine effect. I don't know why you need to check it at all. It is not expected. But at the same time, but what was surprising for me to see that PTH group tend to have a higher uterine weight. Now, why should it be like that? And another issue is that why the PTH group have lower rank ligand compared to OVX? Because PTH actually enhances rank ligand, and that is why it cannot be given after a certain duration, in human maximum 2 years. So why did you see that the rank ligand in the OVX rats given PTH is reduced compared to OVX alone? You have that data. You showed that data. So one, another thing, it is possible that rank ligand alone is not a very accurate accurate expression of the the triad. I know, that the rank ligand, rankel, OPG triad is actually best presented by the ratio of rank ligand to OPG
Yeah,
ratio.
we should have done OPG also, no, in that way. That would have been.
Yeah, so that you could have you would have got a more accurate reflection. But PTH is a direct target of rank ligand. Means PTH sits on the Krebs. PTH responsive, the
Yeah.
cyclically responsive element is a positive, is sitting on the promoter of rank ligand. So when you give PTH, the rank
Yes, I
ligand
know.
will go up. So
Yeah. That is
check.
anti-resolving cytokine. Yeah.
So it becomes that is the reason why PTH is actually
Yeah.
pro-resorptive.
Mm-hmm.
Pro-resorptive. That is why it has that anabolic window that initially the stimulation of formation is much higher than resorption, but with time it catches up. The reason being the rank ligand. Rank ligand is the major culprit.
Okay. Okay.
So I was a little curious to know why you got how do you explain that? Um,
Actually, we were studying the anti-resorptive cytokines, no?
hmm.
Cytokines which are responsible inflammatory cytokines. In that we have selected interleukin 6, TNF alpha, and in that also we have selected the rankel also to study the, because our
Yes, yes,
peptide
yes.
was an anti-oxidative peptide, so we were looking for that. So to answer that, since we are giving PTH, so rankel level should not be that less that you are saying, no? Because it
Yeah,
is,
that is why I'm curious to know why PTH group is showing a
yeah,
suppression of rank ligand. Hmm. So that is what I wanted to know.
yeah, that is
Yeah,
the point. I have to
right.
check. Yeah,
Yeah.
good questions.
Another thing, you found that you said that these ACE inhibitory activity of the peptide.
Mm-hmm.
Now, and you have already said that ACE inhibitors have shown to prevent osteoporosis, probably because the inhibition of renin-angiotensin system.
Yes,
But
yes.
my literature study shows that it inhibits renin-angiotensin, but not necessarily nitric oxide. So now, would you then assume that if you tested your peptide with ACE inhibitory activity in hypertensive rat, it will improve the bone condition, because in the hypertensive rat they have a poor bone condition.
Yes, yes, yes. Yeah, that. But to get that right, it is difficult.
We have spontaneous SHR rat. We have SHR rat. So
Okay.
that's, yeah. We have they are called spontaneous hypertensive rat.
Hypertensive rates. Yeah, yeah.
So we have done a study, and also it is there in the literature in these rats. These are basically in the Wistar background. So
Yes,
in
yes.
the Wistar background, some of the rats which were hypertensive were then bred, and that is how the colony was made. Not by us; it is known in the literature. So compared to the non-hypertensive Wistar, um, the hypertensive rats have a fairly impaired bone mass and architecture. So that is possible. You can check it there. And in line of your 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 thesis that this ACE inhibitory peptide improves bone, does it at the same time promotes the bone vascularity? Because under hypertensive condition, you know, the vasculogenesis in the bone is affected. So the bone vascularity you can also check by these
Thanks,
peptides.
sir. Thank you. Good suggestion.
Okay.
Thank you. Thank you,
Okay.
sir.
Let others ask question. I was asking alone. Please feel free to ask.
Yeah.
Some people apparently left. Is there any more question for Dr. Kapila? If not, then let us join her to thank Dr. Kapila. Join me to thank Dr. Kapila for this wonderful presentation and a very impressive body of work, which we all thoroughly enjoyed. And you are welcome to use our facilities in
Thank you.
future, and for whatever need you find it to be useful, please feel free.
Thank
And
you.
wish you all the best for your future endeavor.
Thank you. Thank you, sir.
Thank you so much for
Thank
joining
you
us.
for giving me this opportunity and
Okay.
interacting with you. Thank you so
Sure.
much.
And another thing that, so as you know, your team has been included in this webinar series. So 2 weeks from now, there will be another talk. So tell your
Yeah.
lab members and others to join. They will get the email and everything.
Sure, sir. Sure.
And please suggest us any potential speaker.
Okay, sir.
Okay.
Oh, thank you.
Okay, Ji. Thank you so much. Bye.
Thank you.